All right. Thank you everyone for joining us today. This is the In Health Precision Medicine Society, a new student-led organization sponsored by the Hopkins Medicine in Health Initiative that aims to explore the benefits and challenges of integrating precision medicine into modern-day healthcare system. Precision medicine is delivering the right treatment to the right patient at the right time. It's not a relatively new concept in human history, as you can see here by the quote from Hippocrates, but only recently have we begun to empirically elucidate the variations in disease response thanks to the advent of next generation sequencing technologies. Throughout the academic year, we hope to set the groundwork of precision medicine following the framework outlined here on the slide. Previously, we've held a talk on genomics, and today we will be holding a talk that will primarily encompass the biostatistics module. Through our events, we want to prepare the next generation of healthcare leaders through symposia, discussion forums, webinars, and more. We also hope to provide a platform for our members to voice their contributions to developments in this emerging frontier in healthcare. Today, we are honored to be hosting Dr. Olivier Elemento, who will be presenting his work regarding the role of AI in precision medicine.
Before we introduce Dr. Elemento, we'd like to take a few moments and share our new blog, the In Health Chronicles. On our blog, we aim to publish short articles about recent developments in precision medicine, making these discoveries accessible to wider audiences. Our first post, Bringing Precision Medicine into COVID-19 ICUs, has been featured by the Johns Hopkins In Health Initiative, the Center for Clinical Global Health Education, and the JHU India Institute. Check out our article using the upper QR code, and if you're a member of our organization and want to contribute to the blog, you can scan the lower QR code to become a member of our blog team. Dr. Olivier Elemento is the director of the Englander Institute for Precision Medicine, associate director of the Institute for Computational Biomedicine, and a professor of physiology and biophysics at Weill Cornell Medicine. His work focuses on big data analytics for precision oncology applications, including gene regulation in cancer cells, computational algorithms for identifying new cancer mutations, cancer epigenomics, and tumor genome evolution. Dr. Elemento developed the first whole exome sequencing test for cancer cells approved by New York State and a recent thyroid cancer detection algorithm. He is the recipient of the NSF CAREER Award, the Hirschl Trust Career Scientist Award, the Walter B Wriston Award, and the Daedalus Award. And with that, Dr. Elemento, the floor is yours.
Great, thank you so much. Thanks for having me. Let me just share my screen and then we'll get started. I have a few slides. I'm hoping I'll be able to get through, at least, most of them. So I'm gonna get started. These are my disclosures. And this is what I'm gonna talk about today. So I'm gonna start with the definition of precision medicine, which I think is important, and I think resonates with some of the things that I've heard in our discussion prior to this talk. Focus on technology. One technology that we use a lot more and more is whole genome sequencing, and we see a lot of potential for it. And I'll try to explain how we can really unleash this potential. But also want to highlight the fact that when it comes to precision medicine, which is, for us, trying to understand really well what's happening in clinical samples, there are many more options now, including the ability to look at tissue at single cell resolution, to almost take snapshots of what tissues look like and how the cells potentially interact with each other and so on. And that, I think, has also great potential to be further used as a way to stratify patients, as I'll try to show you, and hopefully down the line to guide therapy. And if I have time, and I'm not sure well, but I'll try, I'd like to kind of give you a short story about something else, which is kind of mostly on the other side. Thinking about how we can use some of the same techniques that I'll show you today, not so much for therapy sort of selection, but actually for almost designing our therapies. Coming up with new therapies and bringing them to the patients faster than what we've been able to do so far, okay? All right, so I'll get started. So my slides don't always advance the way, okay, here you go. So I get started with a definition of precision medicine, which is, in my view, essentially personalized medicine, but driven by technology. And the reason why I say this is that medicine has always been personalized, as we saw in the quote that you mentioned. But now with technology, we have the ability to personalize medicine in ways that we could only dream of a few years, even a few years ago. Like we discussed, we can sequence somebody's genome in 48 hours or probably even less. In fact, we can sequence lots and lots of genomes in a short amount of time. And we have access to these data analytics tools, such as deep neural networks, that allow us to do really magical things, really analyze gigantic data sets and understand them and predict certain types of outcomes, for example, better than likewise we could even dream of a few years ago. So the tools and the technologies are there. Now it's really how are we going to use those tools in the context of precision medicine. So first of all, when it comes to precision medicine, there's usually kind of a workflow, and that workflow, this is a workflow here that we have for cancer patients, but it kind of really applies to a lot of different patients. The first thing to do is to get samples from patients. If it's a cancer patient, then you have to get a cancer sample. If it's not a cancer patient, typically you try to get some kind of sample that's as connected to the disease as you can. Very often it's a blood sample because that's easy to get. For cancer patients, we have to get both a tumor sample and also a blood sample because we want to understand what's special to the cancer sample, and that's not found in let's say the blood sample. That typically involves focusing on the DNA, but there are lots of things that you can also look at. So it starts with clinical sample acquisition, then profiling, and I'll show you more sort of detail about this. And then there's all kinds of analysis that need to happen. A report needs to be created, and that report needs to be discussed, and then give rise to a highly personalized treatment recommendation. And as I said, this kind of workflow here is really applicable to pretty much every precision medicine kind of application, not only cancer. And so obviously one thing that's important is to have consent from the patients. You want to be able to reuse the data for research, so there's a lot that needs to happen. We have done this process that you see on the screen here in cancer patients for about 2,000 patients, and actually maybe close to 3,000 now. And so we have a ton of data, and we are analyzing the data also for research. The kind of data that goes back to patients after sequencing, we will sequence kind of a core of precision medicine for us is DNA sequencing. So what goes back to the patients and to the physicians is a report that captures the mutations that we found in the tumor cells and not in the normal cells from the same patient. And then we kind of break down those mutations into different groups. There's actually quite a bit of work happening in the field to understand how to tell whether a mutation is really important. Now, actually, most people rely on databases. There's some databases like OncoKB or PMKB that essentially attach an interpretation to mutations that are found very often in the field. And if, essentially, if these databases tell you that a mutation is really important, that mutation will go at the top of a report. This is an example here of a mutation that's very important because there is a drug that can potentially target the product of the gene that has that mutation. Therefore, it goes at the reasonably top of the report. So that's the kind of analysis that are being done across the board in clinical labs to distinguish important mutations, like you see here, from the less important that are a bit below in the report to the basically not particularly important mutations that are at the bottom of the report. All of that is changing very quickly. A mutation that was not important yesterday may actually turn out to be important today as a result of reclassification, a mutation, better understanding of mutations. The field evolves really quickly, and now we understand certain mutations better than what we did before. So we have to be able also sometimes to reclassify dynamically certain types of mutations based on new knowledge that's being acquired almost in real time. Part of the frustration is that, again, lots of mutations that we don't understand in those reports. Mutations that we understand, but that are not targetable. I'll just give you an example, right? So this is a clear cell carcinoma. Many of these clear cell carcinomas have ARID1A mutation. It's a little fuzzy here, but you can see this ARID1A. That's a very common mutation in this tumor type. And in a sense, it helps in terms of diagnostics or diagnosis, right? If you see this mutation, it kind of confirms the diagnosis. But it's not targetable. There's no drug that can target tumors that have these mutations and are going to respond better to a particular type of therapy. And that's something that we see very often. So actually the field in general is, trying to understand better each of these mutations. Typically, that's actually a pretty slow process of, for example, making a mouse that has a particular mutation, seeing if it has a particular phenotype, and seeing what kind of therapies could potentially be effective against the mice that have tumors that bear those mutations. So that's kind of a slow process. There's also a big industry of AI models being created to kind of get a sense of the function of mutations, so that once you know the function of a mutation, you can better target therapies towards that mutation. And I'll get back to this, in the next few slides as well, because that's a big part also of what we do in lots of different ways. One thing that I want to highlight is that the DNA is great, but the field has to go beyond just DNA. I think there's a great future towards analysis of RNA in tumors and cancer, but not only in cancer, across the board for multiple applications. Because the DNA, is good, but it doesn't tell you the whole story. Lots of genes have mutations, but the gene is not expressed, so therefore the mutation can't really be doing anything. Sometimes you have, a mutation, but maybe, I don't know, no downstream consequence of a mutation. One way to actually assess whether a mutation is active or not is to look at pathways that are downstream of a mutation. What we did is actually to build a catalog of gene sets that we know should be active if a mutation is active. And we're actually using a tool like GSEA that some of you may have heard about, as a way to assess whether, if you see a mutation, this is an example here where this is a patient that had a mutation in ERB2, and we have some set of genes that we know downstream of ERB2. So if a mutation is active, those genes should be highly expressed. And so we can assess using GSEA whether that's the case. And we've seen this patient here, this is a positive GSEA result. So it tells you that in this one patient here, compared to patients that don't have this kind of mutation, ERB2 downstream genes are highly expressed as a whole. And that just basically tells us that ERB2 is actually active. And therefore, if you have a drug that targets ERB2, this patient may actually be a responder, because ERB2 is doing what it's supposed to be doing. So just an example of how RNA can be used as a way to credential mutations and give you an additional layer of function that is very helpful when it comes to analysis of individual patients. And this slide here is just to show you that despite the fact that we actually do want to do RNA analysis for as many cancer patients as possible, it actually turns out to be pretty hard because the kind of samples that we get in the context of clinical care is typically kind of highly degraded type of samples. The RNA is degraded very quickly, so we had to optimize certain types of assays to be able to capture expression profiles from tumors. And we actually capture the RNA. The RNA is kind of degraded into short fragments. And so we had to capture the RNA to be able to quantify the abundance of RNAs in tumors. So an example of how we had to optimize the technology to be able to have it applicable to clinical samples that we collect for patients with cancers. But this is kind of what happens when you are able to find a mutation that's important in driving a cancer. That's here a bladder cancer patient, a metastatic bladder cancer patient, as you can see here, with multiple tumors. We did a genome scan. We did a whole exome sequencing scan of this tumor, and we found lots of mutations, but one caught our eye. It was an amplification. This is this little spikes here, amplification of a gene called HER2. And actually, HER2 amplifications are not really particularly well known to be present in bladder cancer, but actually very common in breast cancer. In breast cancer, it's so common that people design a drug to be able to target the product of a mutation. That drug is well known. It's called Herceptin. And I'm sure maybe some of you have heard about this. A drug is extremely effective in patients that have HER2 amplification in breast cancer. Because this patient here had so many metastatic tumors that essentially was going to die as a result of all these mutations and all these tumors. We treated this patient with Herceptin just as a kind of last result. But as you can see, that actually treatment had a miraculous effect. All of the tumors of this one patient here were just essentially destroyed as a result of targeting the cancer with a drug that targets what's really driving the cancer. And so that's a great example of the effectiveness of precision medicine and the ability to scan an entire genome and find things that you don't always expect to find. What's interesting is that these, some mutations keep showing up across different types of cancers. That's a patient with a different type of cancer that also had the same kind of mutation. It's a uterine cancer. The same mutation was found in this one patient here. It's multiple copies of that gene called HER2. So we decided to also treat that one patient with Herceptin, just like the first patients that I showed you. And as you can see here, that patient also had a very, really very durable and stable response, and the tumor basically just melted away. I think that's a really cool example of how you can find the same mutation across different types of patients, different types of tumors, and actually, have a drug that's quite effective across different types of cancers. And honestly, from my point of view, I think that's really the future of sort of cancer medicine. It's not essentially focusing on where the tumor is found in the body. It's actually really focusing hard on what's driving the cancer, what are the mutations that are driving the cancer, and kind of matching drugs to patients based on this information. And as it turns out, there's actually quite a few drugs now that have been approved based on what kind of mutations patients have and not where the tumors are found in the body. These are tumor agnostic or tissue agnostic indication drugs. Pembrolizumab is an example, which has been shown in clinical trials to be effective in patients that have MSI microsatellite instability, irrespective of where the cancer is found. There are other examples, for example, larotrectinib, which is shown to be effective in most patients with NTRK gene fusions, irrespective of where the tumor is found in the body. So, this kind of tissue agnostic definition of cancer is really, I think, the future of cancer treatment. As I told you, we do enroll patients into research protocols, and so we can do all kinds of analysis on the tumors once we have the tumors. And also, we actually, for example, what we do is to look at multiple time points. This is an example of a study in bladder cancer, looking at the comparison of mutational content between metastatic tumors and non-primary tumors, the first tumor that was found in the patient across different patients. One thing that we find by comparing the list of genes that show up across the primary tumor and the metastatic tumors, we can essentially build phylogenetic trees of tumors, like almost trees of evolution of the tumors, looking at how many mutations are shared and how many mutations are specific to each sample, each site. One thing that's really interesting is that you see across many patients that the metastatic tumors, which is those black dots here, and the primary tumor here, are actually quite different from each other. They basically share a few mutations. These are the branches here in red, but they actually have evolved across different trajectories. They acquire different mutations, in different ways. And this is something that we see, as you can see here in bladder cancer, quite a bit. But it does tell you something fundamental, which is that when you have a patient with metastatic disease, if you do a biopsy of a patient, right, you can't really expect to find the same mutations that you had much earlier in the kind of life history of the patient. So you really have to sequence the patient as you see the patient, as opposed to relying on information from the past, which is unlikely to be reliable based on this kind of analysis. Sorry. Yeah, so this is to kind of give you a sense of where we are in terms of precision medicine. As I say, the future of precision medicine for us is whole genome sequencing. What I've shown you so far was called whole exome sequencing, when you only sequence the genes. And that's just giving you a very partial picture of the genome. We started a very large project at Cornell focused on whole genome sequencing, where we're sequencing now thousands of patients across different indications using whole genome sequencing, where basically we sequence the genome from beginning to end. The genes and also what's between the genes, and we analyze this data to try to get more insights into what's happening in each patient. Why do we like whole genome sequencing? Because essentially we think that one assay can replace lots of different assays. We can get a lot of information out of whole genome sequencing. It can help us diagnose conditions that are hard to diagnose. It can replace multiple types of testing with one test. It can allow us to look at different types of signatures and confirm, for example, that certain mutations are actually functional. You can do pharmacogenomics, which is to look at certain genes that are involved in the metabolism of drugs and ask whether, which patient has which gene, and maybe sort of tailor the dosage of medication according to the genome. And you can do things that I'm going to talk about next, which is to calculate polygenic risk scores, which is essentially a prediction of risk of developing certain types of conditions. which I think is also a great tool that I think people are going to be using more and more in the future. In the context of cancer, we've shown actually that you can get more information out of whole genome sequencing compared to whole exome sequencing. With whole genome sequencing, this is a busy slide here, but you can actually find all kinds of mutations that are pretty cryptic when it comes to whole exome sequencing, including what we call structural variations and complex structural variations, where you have essentially translocations, kind of breakage of the genome into small bits and kind of reshaping or reformation of a new chromosome with essentially kind of combination of those different bits. That's a phenomenon called chromotripsis that we actually see in quite a few patients. This is kind of looking more locally, but you see here, this is a complex structural variation that's affecting a gene that's very important in lung cancer, which was the subject of this paper here. That's a kind of complex mutation here that would not show up using whole exome sequencing, but does show up quite clearly using whole genome sequencing. And so that's just an example of how getting more juice, if you want, out of whole genome compared to whole exome. As I said, we are using whole genome sequencing across the board for many, many different types of applications, not only cancer. We've actually published quite a few studies, for example, in spina bifida, which is a kind of developmental defect that we've shown is also caused by a combination of structural variants and point mutations, likewise, where whole genome was able to illuminate the causes of a disease in a patient-specific way, in a way that we couldn't do using traditional methods of sequencing. One cool thing also where we can use whole genome sequencing is a way to calculate prediction of risks of developing certain conditions. One thing that we did a few years ago was to look at thrombosis. Are we able to predict who's at risk of thrombosis, which is basically blood clots using the genome? One thing that we did is to look at all kinds of clinical factors, and lots of clinical factors are connected to risk of developing thrombosis, for example, age or BMI and so on. But one thing that we can also do once we have a genome is to actually ask whether a patient is at risk of developing thrombosis because the patient's inherited a lot of polymorphisms in the genome that in genome-wide association studies done in the past have been connected to thrombosis. As you may know, people have done these genome-wide association studies across different types of disease. They typically found SNPs that increase the risk by a small amount. But what happens is that some individuals inherit, let's say, 20 SNPs connected to increase in risk of the same disease. So each SNP increases risk by a small amount. But if you get 20, it kind of adds up. It actually kind of add up to pretty substantial amount of risk. So we can actually, polygenic risk scores is about using all of these different SNPs as a way to kind of add up and calculate the actual risk of developing certain conditions such as thrombosis. One thing that we showed in this paper that we published last year was that you can actually predict risk of developing thrombosis by combining clinical factors and genetic factors better than each one taken by themselves. So the combination of genomics and clinical factors is the most powerful in terms of prediction of thrombosis, and to the point where the accuracy of prediction is actually quite good, and you can actually potentially use that kind of tool in the clinical setting as a way, for example, to do prophylactic type of medication for patients with high risk. If patients come to get surgery, right, it kind of makes sense to actually have a genome sequence and look at the clinical factors to predict whether they're at high risk of developing thrombosis after surgery, and maybe give them prophylactic anticoagulants as a way to mitigate those risks. And this is again something that we are showing here. And in fact, Scott in my lab has shown that across the board, adding a genomic component to clinical factors, which often used to predict risks, actually does increase the ability to predict risks in almost all disease. For 21 out of 23 diseases that we looked at, if you are using clinical factors, you can predict risks of developing those diseases. But actually adding genomics does increase your ability to do so by a substantial amount across the board. So that's a great demonstration of the utility of genomics. And the reason why that's important in the context of whole genome sequencing is whole genome sequencing, we sequence the whole genome, right? So those SNPs that come up in GWASs, they're actually often outside of genes. So if you only sequence the genes, you're going to miss those SNPs that are found in GWASs. And therefore, this is something that is revealed by whole genome, but it's very precious and very useful for example, for predictions of risk of developing conditions. One of the things that we showed also in that paper that we're still looking to publish now is that there are actually quite a few differences in terms of predictive power for these polygenic risk scores, depending on how you look, essentially which different populations you look. So for example, it turns out that, in some cases, that difference is in the ability to predict risks, depending on whether you're female or male, sorry. As you can see here. Sometimes there's a difference in predictive power if you're, young versus old, as you can see here. Sometimes there's actually a difference in predictive power, and if people belong to different ethnic groups, as you can see here. So these are kind of things that we need to keep in mind. The predictivity of these scores is not equal across different groups. So we have to be able to understand how to use them to hopefully be able to use them in a clinical setting. We're doing quite a bit of analysis, outside of genomics. This is an example of a study that we are doing now, looking at prediction of progression from pre-diabetes to diabetes. We're doing this using metabolomics, which is the prediction, essentially the quantification of metabolites, inside of metabolites in the blood, which can also be done now actually very accurately using NMR, using different types of technologies. Nick in my lab was able to find that pre-diabetic patients, if you look at the metabolomics of the blood, the metabolite levels, across hundreds of metabolites, you can segregate pre-diabetic patients into 7 different groups. And if you look at the tendency of these groups to progress to full-blown diabetes, there's actually quite a bit of difference between the groups. So the metabolites in the blood are also predictive about progression from pre-diabetes to diabetes. And I think that's also likewise a tool that we are hoping to be able to use at some point in the clinical setting. And Nick has built some kind of app to be able to hopefully help patients and physicians kind of use this kind of information as a way to navigate the prediction of risk based on metabolites, but also things that could potentially be done as a way to maybe mitigate the risk of progression. So switching gear a little bit, I wanted to maybe give you a little bit of information about another technology that we use for tissue analytics. This is very orthogonal to the genome. This is really trying to get more information from clinical samples. We're looking at not genomic profiles, but looking at tissue architecture, which I think is really also really important. We're using a technology called imaging mastodometry, which is essentially antibody-based analysis of tissue. Antibodies recognize different markers that, for example, are specific to different cell types or certain things happening within cell types, which you can use as a way to stain tissue with lots of antibodies at the same time and get kind of a multiplex sort of view of tissue. This is a single cell resolution analysis, and therefore you can really sort of see the cells in this kind of analysis. This is what it looks like here. This is the kind of tissue that goes into the machine. This is here a few slides regarding one project that is quite large scale that we're excited about, to use this kind of technology to analyze B-cell lymphomas. So these are cancers of B cells, cancers of immune cells. We've been looking at hundreds of these tumors and analyzing these tumors using panels of antibodies, using the technology that I showed. These are the kind of antibodies that we use here. And the idea is to mark different types of immune cells, as you see here, different types of functional kind of markers across the immune cells, as well as look at some structural markers that, for example, can highlight where blood vessels are in the tissue. This is what it looks like if you want to look at only a small number of channels. With this kind of technology, you can only look at 4 or 5 channels maximum at the same time, because otherwise the colors kind of bleed into each other. But each of the colors, you can see the cells, little blobs here in this kind of picture here. And you can see, for example, things like blood vessels, which basically holes through the tissue. And then you can essentially see that you have different cells expressing different markers. These are what individual tumors look like. Each of these round sort of disc here is a little punch within a particular tumor, and you can see the cells and the diversity of cells, and things like blood vessels. Like I say here. So what we have to do is kind of technology here is to essentially find the cells, segment the cells, and then assign a kind of essentially quantify the markers within each of the cell, and then essentially assign a function to each of the cells. That's how we're able to find the B cells, the T cells, the endothelial cells, the macrophages. That's basically based on finding the cells and then analyzing which markers are expressed within the cell. It does tell you what the cell is. And we do this across, this is an analysis here that included 7 million cells. So this is a very large analysis. So when we do that, we are able to cluster the cells into groups of cells that express the same markers. And this is what you see here. I think we ended up with about maybe close to 35 different cell populations. Basically, these are cells sometimes across different tumors that express the same markers. So you see, this cell population here, each row is a cell population. This cell population is expressing a very large amount of these 3 markers here. This is CD163, CD14, CD68. And so that's because we know that CD68 is a kind of macrophage, that just tells you what this marker is all about. And so this is what happens once we're able to identify, go from essentially a cell to an identity. Each of the cell now has its own color here because we know exactly what's the identity of a cell. So there's, for example, the green cells are B cells. So one thing that we can do, and actually is quite unique once you have this kind of information, is to actually, for example, forget about the tumor cells, which are mostly the B cells in this kind of cancer, and maybe quantify the extent to which you can cluster the tumors into different groups based on only focusing on the non-tumor cells, focusing only on the microenvironment, the immune cells around the tumors, which is obviously an interesting, maybe unusual sort of type of approach. But we figured, maybe, I don't know, there's something important about the microenvironment of tumors, and maybe we can quantify and say categorize tumors into different types of microenvironment. And it turns out when you do that, actually, you can tell that there are very different types of tumors. There are tumors that have, for example, very few immune cells. These are these tumors that we call cold here. Tumors that are very rich in immune cells, as these are the cytotoxic tumors here. But if you look at what tumor cells are highly abundant in this kind of cluster here, you see there's actually a lot of dysfunctional immune cells. So these are dysfunctional macrophages here that express immunosuppressive markers, dysfunctional T cells that express also immunosuppressive exhausted markers. So basically, immune rich but dysfunctional. And then in between, you have the other types of clusters, CD4, inflammatory, which are kind of a little bit more like what you see in normal lymph nodes, which is kind of the normal counterparts of these B-cell lymphomas. So it's actually really cool that you can cluster these tumors into 5 different clusters. This is an example illustration of these different clusters here. This is an additional analysis that we did to credential better each of the clusters. I'll actually move quickly on this. A pathway analysis of each of the clusters. But what's really cool is that then we actually looked at survival, which is essentially clinical outcomes. All these patients get the same therapy. One question is whether there's differences in terms of survival based on the patient falling in one of these different type of clusters only defined by the microenvironment. And what's really cool here is that if you see, right, so these patients here, the cytotoxic patients, these are the patients that have a lot of dysfunctional immune cells in the tumors. These are the patients that do very poorly when it comes to chemotherapy response. Now, what's interesting is that if you look at the second group in terms of worse survival, these are those cold tumors, those tumors that have no essentially immune cells in their tumors. Which is interesting because basically what it kind of tells us is that tumors, to be able to respond to chemotherapy, they need to have almost like a healthy immune system. They need to have healthy immune cells. If they have these very messed up immune cells that are completely immunosuppressive, very essentially dysfunctional immune cells, you don't respond to chemotherapy. If you have no immune cells, you don't respond to chemotherapy. So you need to be in between to be able to respond to chemotherapy. And we think that's a pretty profound insight, not only in terms of being able to stratify patients into different groups, but also in terms of potentially, thinking about strategies to reestablish a normal immune microenvironment for tumors. There are actually quite a few drugs now listed, candidate drugs that you can potentially use to target the cytotoxic clusters here to get rid of the dysfunctional immune cells and maybe sensitize those patients to immune therapy. So these are some areas that we're exploring now. One more thing is that, as I told you before, we're very interested in finding the function of mutations. It's actually something that you can do with this kind of data because you essentially get a very detailed map of what's happening within a tumor in a special sort of way. You can correlate the presence of mutations in those tumors, which we can calculate, which we do randomly, we do routinely, as I mentioned sort of before. So for example, one thing that we see is that tumors that have mutations in B2M, which is a component of the HLA complex, they actually tend to have lower expression of HLA as a result of the B2M mutation. So, if you didn't know that B2M was connected to HLA, actually it would be a way by which you could tell that it is. And so, in this way, we're actually able to build maps of the potential consequence of having a mutation on the microenvironment and what's happening in the tumors. And that actually does give us a tremendous amount of hypothesis regarding the functional mutation. It's like essentially kind of a functional map of mutations that you get from this kind of analysis here. So lots of hypotheses coming out of this. All right, so I'll keep going. I just want to give you one more story, and I'll try to make it short. This is, as I say, the bit orthogonal. This is how we've been able to use AI as a way to help bring new molecules to the clinic faster and improve likelihood of clinical success. This is here really based on this idea that we have lots of drugs nowadays, lots of candidate drugs, but by and large, for many drugs, we don't quite understand what they do in human cells. That's something that we, in my lab, we've been focusing on for some time, finding the target of molecules that don't have a target. We've used all kinds of different methods for doing this. But recently, what we've done is to actually try to use the power of machine learning as a way to connect small molecules with the targets within cells. And long story short here, we've been doing this using a method that's focusing on data integration with the idea that on molecules, you can measure quite a few things nowadays pretty easily. For example, what you can do is to take cell lines, treat the cell lines with the molecule, do gene expression profiling before and after treatment, and see what genes go up and down as a result of the treatment. That does not give you the target of the molecule, but it potentially gives you some idea about the pathway in which the molecule is found, right? So not a target, but maybe something close to the target. You can look at side effects as well of treating let's say mice with a molecule and different side effects, and that also does give you a little bit of information about the target. You can look at cell line viability across different panels. You can look at the structure of the molecule. All of these information bits do give you a little bit of data about what the target is, but it doesn't give you the whole target. We created a method that's using a Bayesian network, a Bayesian kind of simple Bayesian network to actually be able to integrate all these different data points to predict the target of a small molecule. It's not a structural biology approach. It's really based on purely data and machine learning. And as you can see here, when you have actually quite a bit of data about a molecule, the combination of those different data sets here, you can actually predict what's the target of a molecule in a very effective way, close to 90% efficacy. So that actually works really well, and we've been able to use this across the board. One thing that we've been able to do is to actually screen a large number of small molecules and predict targets for these small molecules. So we've been able to do this actually in about 40% of small molecules that did not have a known target before. Once you do this, you can actually look at specific predictions that are potentially interesting. One of them was looking at microtubule inhibitors. There's a whole industry in cancer of microtubule inhibition. This is a very effective type of therapy in cancer. But a lot of patients actually become resistant to microtubule inhibitors. And so there's actually always interest in finding novel microtubule inhibitors. Using this analysis, we were able to predict microtubule inhibition activity for 24 molecules in our huge database that you see here. So we actually went to the bench with our colleague here at Cornell and actually showed that indeed we're using additional assay, biochemical assay, 14 out of those 24 predictions actually turned out to be real. and we're indeed able to actually destabilize microtubules, as you see here, which is quite amazing. These are like novel structures, new molecules that were not known to have a microtubule inhibition activity that we predicted to do so, and actually turned out to have such activities. But what's even cooler is that we actually found among those 14 at least 3 or 4 microtubule inhibitors that are effective in the setting of resistance to every known microtubule agent. So these are basically structures, molecules that have a structure that's very different from everything that's known now, and therefore is able to be effective in the context of resistance to traditional therapies. So as you can imagine, very exciting molecules to potentially counteract resistance to this to microtubule inhibitor resistance. But we could do more things. One thing we actually did was to work with a company out of Philly on a molecule that they had that did not have a known target. It came out of a phenotypic screen, did not have a target. Was able to kill cancer in some settings, but they didn't really quite understand why and how that would happen. Because they didn't have a target, it was very hard for this molecule to actually be further developed. I just didn't know what to do with this molecule. So we applied this AI and machine learning technique that I showed you to this molecule here because we had a bit of data about it. And we're able to make a prediction that was actually quite unexpected, which is that this molecule, according to our prediction, would be able to target dopamine receptor 2, which is not a very typical anti-cancer target. It's actually very atypical. But the company was actually kind of a little bit desperate, and they really wanted to do something with this molecule. So they actually tested this prediction that ONC201, this molecule, was able to target DRD2. And as it turns out, all of the experiments that they did actually indicated that indeed our prediction was real. They did binding experiments, showing that indeed ONC201 binds to DRD2. They did activity experiments showing that ONC201 is able to disrupt the downstream kind of activity of DRD2. And also because this molecule actually had gone in patients in a small clinical trial that failed, they actually were able to measure certain things before and after treatment. And prolactin is actually known to be downstream of dopamine receptors, DRD2. And as you can see here, patients after treatment have an increased level of prolactin. So all of these different experiments showed that indeed DRD2 is the target of ONC201, which is actually really cool. It's like a machine learning method that was able to make a prediction that led to essentially a novel finding. And what's even cooler is that as a result of this prediction here, the company was able to reposition its clinical trials towards patients that express a lot of DRD2. For example, H3K27 methylated gliomas are actually patients that express a ton of DRD2. The company started clinical trials in this patient population here, which is extremely hard to treat. This is was very sort of difficult to treat sort of type of brain cancer in children and young adults. But actually, what's really amazing is that this molecule actually turns out to be effective in these patients. Actually, a lot of patients were responding to this agent, including there was some success stories that were highlighted as a result of some patients really getting a miraculous almost cure as a result of this therapy. So this is really, I think, from my point of view, as you can imagine, a great utilization of machine learning and AI to kind of do something that is actually really benefiting patients. Including the patients that are extremely hard to treat, and then I think really a great story. And now we're actually doing more experiments. We actually found another tumor type, a subtype of metastatic prostate cancer that we actually realized also has a tremendous amount of DRD2, and we actually started treating cell lines with ONC201 that belong to this metastatic prostate cancer subtypes. And we got actually a great efficacy for being able to kill cells of that cancer subtype. We actually did also a mouse experiment showing that indeed, this ONC201 molecule is able to actually effectively kill in the mouse human tumors growing in the mouse, with PDX sort of models. So again, I think a great potential to use this therapy, this drug here, as a way to treat patients that have this biomarker of this DRD2. And again, if we had not used an AI model to predict that DRD2 is the target of ONC201, we would never know that we should position this drug into this particular indication. Now there's so many indications, and this is a very niche and a very small one. So it's a great story in my view, and I think a story that we hope to be able to repeat multiple times. And that's the end of the talk. And I'm be happy to take questions.
Yeah, thank you very much, Dr. Elemento. I guess, does anyone have any questions to start us off?
Yeah, thank you so much. That was very insightful talk. So I had a question about polygenic risk scores.
Yeah.
And just broadly speaking, how do you see them being implemented into the clinical setting? How do you think they could help cancer patients, for instance, or also, other, chronic disease, care?
Yeah, that's an excellent question. And I think, the answer to this, I think in my view, is really that we have to think about this kind of genetic testing and screening in a slightly different way. So one of the things I didn't really have time to really go into details about this is our vision for sequencing, here at Weill Cornell is actually a very kind of, ambitious vision. Part of the issue about genetic testing in general is that right now, when you do this in the context of clinical care, and that's something that you may have been exposed to, it takes a very long time before the time you order the genetic test and the time you get the results. Our vision is a bit different. We are saying at some point, we're hoping that every patient, when they come into this hospital, will have a genome sequence right away, right? If they come because they have, let's say, an allergy, right? We actually have a genome sequence. We do the genome sequencing in a clinical lab, but we actually put that's the trick. We put the genome in the medical record, right? And so the genome can be used throughout the entire life of a patient. So if at some point we want to be able to predict whether a patient is going to have thrombosis because the patient is about to get surgery, whether the patient is at risk, like we have the data that's already there. We don't have to wait. We don't have to ask the patients to wait a month to get a report to be able to tell the patients, oh, you have a high risk of thrombosis. I think this integration of genomics into the medical record and the ability to reuse the genome throughout the life of the patients because it's already there and not something that you need to acquire is, I think, instrumental towards the adoption of this kind of testing and this kind of utilization across the board. For cancer, there are some types of polygenic risk scores that are actually quite effective. One of them is, for example, prostate cancer. Prostate cancer can actually be, risk of prostate cancer is actually quite predictable from polygenic risk scores, as you can see here. Especially if you have, essentially kind of combination of clinical factors and genetic factors, as you can see here. And so, it's actually predictive to the point where you can use it as a screening mechanism. So, imagine that, everybody gets a genome sequence when they come here into the hospital. We could immediately tell somebody ideally somebody young, right, that, in the future, you have a pretty high risk based on your genes to develop prostate cancer. So the question is then what can be done, right? So, one is obviously certain type of lifestyle alterations that can potentially mitigate risk of developing prostate cancer. Additional screening, maybe more frequent screening, right? So there are ways to screen for prostate cancer now using a blood test that's measuring PSA levels. That's typically done every other year, or maybe every year above the age of 50. If I had a PRS, a polygenic risk score that tells me that I am in the high risk group of developing prostate cancer, I would want to get tested maybe starting at the age of 35, as opposed to the age of 50, right? So this is how you can potentially implement, this kind of testing. But as you can see, there's a lot of steps, right? So one is, ability to use the medical record as a place where you store genomic information, ability to use it, frequently and immediately, and also kind of having a medical system that is able to support what comes after the prediction and is able to, sort of potentially give patients an idea of what to do based on the prediction. So some scores, may not be very helpful if there is nothing that can be done. But some scores like, prostate cancer, I think, really are, really helpful because if you're at a high risk, you can do things like more frequent screening for prostate cancer, and that could be quite effective as a way to detect prostate cancer early. And, as you know, in every cancer, the earlier you detect cancer, the more likely you are to be able to treat the cancer successfully.
Great, thank you. I guess I have a question about, well, you mentioned a lot about how like whole genome sequencing is very powerful in providing us with a lot of data and a lot in terms of the ability to predict and characterize disease. So I guess if we think about whole genome sequencing as being able to do so much, then what does that mean for the role of, say, transcriptomic or like proteomic studies? How do you see all of these fitting in together in the big picture?
Yeah, I think the future is towards this kind of multi-omic type of profiling. The reason why people like DNA in general is that first, there's a lot of information in our genes and a lot of information in the cancer genome, because cancer is a disease that's caused by mutations, right? And so being able to find what's different between cancer cells in terms of their DNA and normal cells, that basically gives you the blueprint of cancer. The challenge is that it's like a blueprint, but it's like I don't know, it's like a giving you lots of pieces of a puzzle that contain a very nice image. Like it's actually not so easy to kind of put the puzzle back together. In fact, very few people are really able to do it, right? But the reality is that the image is beautiful, and once we're able to put the pieces of puzzle together, we'll really be able to understand cancer and treat the cancer in a very kind of patient-specific way. Just it's going to take some time, because of the fact that we don't have enough drugs, we don't understand how to put, the puzzle back together and so on. But, for cancer, I mean, this makes a lot of sense, because that's how cancer comes about. It's because of mutations. There's nothing else, the cancer is only caused basically by mutations. And so the problem is that, as I said, cancer is complex, right? And so, and to some extent, every cancer is different. So being able to just look at DNA and be able to kind of piece together the puzzle is going to be very hard. And I think the idea is that RNA and protein, for example, could actually help you a lot, because you kind of know once you see RNA and protein levels, maybe this pathway is active, this pathway is not active. So now you can suddenly understand the DNA better, and it's kind of what I was trying to highlight at the beginning of the talk. So I think there's great future for a combination of DNA and RNA and protein. The challenge is measurement. DNA is like extremely easy to measure. DNA is super stable. DNA is like the most stable analyte ever. Like you can actually get DNA from mummies, right? From like 2,000 years ago, and actually sequence the genome. And this is like this is like a DNA that just left on the table, like left on in a sort of tomb, and then you can actually recover and sequence. Extremely stable. RNA is extremely unstable, and protein is actually great, but also is actually very hard to measure. Technologies that we use now to measure protein activity or protein quantity are very, very basic in some ways compared to our ability to sequence DNA. And we just, for example, need a lot of protein to be able to assess protein levels. I think that's going to change. I think, there's going to be probably some kind of revolution at some point, some kind of technical breakthrough that will allow us to quantify protein levels, in the same way that we sequence DNA and can do this very routinely. But because that's not the case yet, proteomics is just not used as much as DNA sequencing. So it's kind of a practical sort of thing. It's like DNA is super easy, and it's used across, it can be used across the board. RNA is a bit harder. Protein would be amazing, but it's actually super hard to measure. But the future is towards at least combining those two things, or those three things together, because then you get a full picture of the disease.
Got it. That makes sense. Thank you. And then I have another question about using AI to predict drug targets, like what you showed with ONC201. So do you think something like this could eventually be integrated into the drug screening process, like on an industrial wide level? And if so, like how long do you think it would take for us to get to that point? Because I think that would really be revolutionary.
Yeah, I completely agree. I mean, I think this is actually, so I started a company that is actually basically doing, that is kind of trying to essentially commercialize, some of these insights. And part of the challenge is really kind of, we still need to build the data on these molecules, right? So the structure only, which is easily accessible, does not tell you the whole story. So we need to have the side effect, data that I showed. We need to have maybe cell line data. We need to have a little bit of additional data. And so what we need to do, I think, in industry is just, be able to characterize molecules with this kind of additional simple to acquire data set, so that we can have a huge database of information that we can mine, and then come up with very sort of systematic approaches to kind of connect targets and molecules, build a better machine learning model essentially, and kind of compared to the one I have. The one challenge about this machine learning approach that I showed you is that it's only able to interpolate. So for example, it's predicting connection between molecules and targets, but it's learning from many examples where the molecule that's able to target a particular gene, a particular target. And so if a target is not in the database, then obviously our machine learning method is not able to predict that any drug can target those targets. So that's a bit of a limitation of the technique. But again, there's a lot happening in the field now in terms of using machine learning across the board for many different types of problems in drug discovery. We've actually published papers on, for example, predicting whether a molecule is toxic in humans before doing clinical trials, using analysis of many clinical trials, the ones that failed, the one that didn't fail, being able to understand what distinguishes between them. We've been able to use machine learning as a way to predict whether two drugs, when you combine them together, they are able to kill cancer cells better than each of the drugs individually. It's just examples of many kind of sort of problems that you can address using machine learning, and there are many of such problems. And so my dream is to kind of, the company that I started is almost trying to reinvent drug discovery driven by machine learning, as opposed to the way that we've been doing this for many decades now, which is driven more by intuition and by reading a couple of papers, very often, many of which are not quite right, sometimes even completely incorrect. So we're kind of learning from data to be able to produce insights that we are hoping will guide us towards targets and molecules that are going to have efficacy in specific disease, but the whole process being driven by AI. So we'll see. It's a bit too early to tell whether that's going to work. Maybe it's a kind of a pipe dream, but we have started a company to really address exactly that question.
Got it. Thank you. Yeah, that sounds really exciting. I guess anyone else have any other questions?
I guess to add to that, it sounds like one of the challenges of, accomplishing such a task such as that is that one issue, for instance, is for cancer is recurrence, right? And that even if, say, you target a specific set of subclones, sometimes you get new ones, right? And then that can be a challenge is even if you develop something that targets a specific set of subclones, then you have other ones that may evolve certain mutations that allows them to develop resistance. So I was wondering if that's something that you have looked into, and is that something that can also be tackled using machine learning approaches?
Absolutely. I'm actually completely convinced that's the case. As I mentioned to you, I have actually a couple of students now, and we've done this for a while, using machine learning as a way to predict how to combine drugs. And the answer to your question about how to tackle the complexity of cancers and basically the subpopulations of cancer cells is really, honestly, drug combination. There's actually no other real, approach, for doing this. Maybe a slightly different approach would be to kind of teach your immune system how to recognize, cancer cells in general. That's, people are trying to do that. That's hard. But I think the more effective, way would actually be to essentially target, tumors with different drugs at the same time, be able to eliminate, different cell populations by, essentially targeting them with different drugs. And in fact, even targeting individual cell populations with different drugs so that these populations can't evolve resistance. Some of you may be familiar with HIV and how we are treating HIV now. It is using drug combinations, and the goal of these drug combinations is to prevent resistance from occurring in HIV. Basically, it's like, you corner, HIV in so many different ways that it cannot evolve resistance at the same time. You can't have, and I think it's the same with cancer cells. It's very difficult to evolve resistance to 4 or 5 drugs at the same time, because you have to make like 5 mutations in completely different places. And statistically speaking, that's basically impossible. So that's why, combination therapy works so well in HIV, because, HIV is not able to evolve resistance so quickly. and therefore is targeted by at least one drug. And I think the same thing needs to happen in cancer. The future of cancer therapy is not to treat cancer one drug at a time. It's probably 4 or 5 drugs at a time. The challenge is that everybody's different. HIV is actually a very small virus that doesn't change too much at the end of the day. Every cancer is amazingly different. So to be able to predict which combination to give to one patient, I don't think we can avoid machine learning. I think we need to essentially kind of integrate proteomics, expression profiling, genomics, and essentially predict the combination of drugs that you should employ on each individual patient. In my view, that's actually the future of cancer therapy. It is some kind of a machine learning based prediction of combinations of drugs that are going to be effective in each individual patients to be able to target the different subpopulations and prevent resistance. There's no way that we can do enough clinical trials or pipette fast enough that you can actually test every combination of 5 drugs. In cancer, there's probably now let's say 150 drugs, right, in total. Imagine the number of combinations of 5 drugs that you can get out of 150 drugs. Like, there's no way that you can pipette fast enough to test all of them. You have to use a machine learning approach that's going to tell you choose best in this in combination, because you have this kind of proteomics pattern or this genomics pattern or this transcriptional pattern.
Thank you. I guess anyone else have any other questions? All right, well, if not, thank you again, Dr. Elemento, for presenting your talk and answering all our questions.
My pleasure.
Yeah, we had a great time listening to what you had to say and learning about really what precision medicine is going to look like in a few years down the road.
Cool. Well, thank you so much.
All right, you're welcome. Thank you again. All right, take care. Bye-bye.
All right, take care. Bye-bye.
Thank you.